Welcome, curious minds, to the deep dive. Today, we're embarking on an incredible journey, not across continents or through time, but into the truly minuscule, the nanoscopic world. This is the intricate realm where the fundamental building blocks of life, molecules and proteins, perform their most vital and delicate processes. Imagine for a moment being able to witness this intricate molecular ballet, not as a blurry frozen snapshot, but as a vibrant, live, and dynamic performance unfolding in real time right before your eyes. It's a vision that has captivated scientists for decades, and to truly appreciate that vision becoming a reality, well, we need to unpack a groundbreaking technology. For years, super-resolution microscopy has been a monumental game changer, shattering the long-standing barriers that prevented us from seeing beyond the classical limits of light. These techniques have allowed us to peek into previously invisible realms, revealing the what and where of cellular structures with stunning clarity. But even with these breakthroughs, a new frontier was calling, a leap forward promising to revolutionize not just what we can observe, but how fast and how dynamically we can see it. Today, we're diving deep into event-based single-molecule localization microscopy, or event SMLM. Our mission is to unravel how this innovative technology allows us to overcome previous limitations, particularly in capturing the real-time dynamics of individual molecules. And importantly, why that's not just a technological marvel, but a monumental leap for science, destined to reshape our understanding of health and disease at the most fundamental level.
And what's fascinating here is that we're talking about a technique that doesn't just incrementally improve upon existing methods, it fundamentally rethinks how we detect light. And in doing so, it significantly pushes the boundaries of what we thought was even, physically possible. It offers us unprecedented clarity in both spatial resolution, how finely we can distinguish individual components, and crucially, temporal resolution, how quickly we can observe changes. And this isn't merely a theoretical concept like a clever idea confined to a whiteboard. The rich source material we're exploring today, primarily a cutting-edge research paper from the Indian Institute of Science, Bangalore, it details not just the conceptual framework, but the successful development, practical realization, and robust demonstration of this event SMLM system. It's a proven tool, and its results are truly compelling, offering a new lens through which to view life. So if you've ever found yourself wondering how scientists peer into the living, bustling environment of the cell to watch proteins move, structures change, or signals propagate, then this deep dive is going to provide you with some incredible aha moments about how we're finally moving beyond static, almost archaeological observations to truly dynamic, living insights.
Yeah, moving beyond those static pictures. To really get why event SMLM is such a big deal, we should probably first talk about the hurdles scientists faced for, well, centuries, really. For a long, long time, microscopy hit this seemingly impenetrable wall, a physical limitation dictated by the very nature of light itself. It's called the diffraction limit. Think of trying to photograph a tiny, incredibly fast-moving bird from miles away. No matter how powerful your lens, no matter how much you zoom in, there's a point where the image just becomes a blurry smudge.
Right, it's a fundamental physics problem. Light behaves like a wave, and waves spread out. So when light from a tiny point source passes through your microscope lens, it doesn't focus back down to a perfect point. It creates this fuzzy blob, an airy disk.
Exactly. And that fuzziness means you fundamentally can't resolve two objects that are closer together than about half the wavelength of the light you're using. For visible light, that's typically around 200, 250 nanometers. It's like having a perpetually blurry picture no matter how hard you try to focus.
And that was a huge barrier, because so many critical things inside a cell, individual proteins, molecular machines are much smaller than that limit. They just blurred together.
Right, you couldn't distinguish the individual players. Then, in the early 2000s, this revolution happened. These groundbreaking super resolution techniques just burst onto the scene. You hear names like STED, Palm, Storm. They weren't just improvements, they were monumental breakthroughs.
Absolutely. They found these incredibly clever ways to sidestep that diffraction barrier, essentially tricking the physics of light. Suddenly, scientists could resolve features down to maybe a few tens of nanometers, sometimes even less.
And collectively, these are often called single-molecule localization microscopy, SMLM, right?
Right.
Because they rely on detecting individual molecules that are made to blink on and off.
Precisely. They pinpoint the center of each blinking molecule one by one and build up a super resolved image over time. Incredible tools. They really changed the game for seeing structure.
They gave us this amazing high-resolution map of the cell's interior. But here's where it gets really interesting. The catch. While these techniques gave us incredible spatial resolution, the what and where, at the nanoscale, there was still a big piece missing. We knew where the actors were with amazing precision, but we were still missing the plot, the when, and how fast of their
That's
interactions.
the critical point. If we connect this to the bigger picture, so many crucial biological processes are fundamentally dynamic. They're constantly moving, changing, interacting. Think about proteins assembling into complexes, viruses trafficking through the cell, signals propagating along membranes. These things happen fast, really fast.
And traditional SMLM, because it relies on these standard cameras taking pictures frame by frame, it just couldn't keep up.
Exactly. Even if you get amazing spatial detail, say 15, 30 nanometers, if the process you're watching happens in milliseconds and your camera takes an exposure or a frame every 30 or 60 milliseconds, well, you're missing huge chunks of that action. It's like trying to understand a conversation by only hearing every 10th word. Yeah. You get fragments, but not the flow.
Right. You miss the actual dynamics.
Imagine trying to understand a really complex, fast dance routine just by looking at a few still photos taken seconds apart. You'd see the dancers' positions, maybe get a sense of the formation, but you completely miss the movement, the transitions, the interactions, the speed.
Who wouldn't see the dance itself?
That's essentially the challenge traditional SMLM faced for live cells. You weren't watching a molecular movie, you were looking at a flipbook with most of the pages torn out. So many questions about rapid molecular kinetics remain just out of reach. We could see the actors, but not their performance.
Okay, so the map was great, but the action was too fast. How do we solve that? How do we get the map and the real-time action? This brings us to the core innovation, the event camera. This sounds like a fundamentally different beast compared to normal cameras.
It really is. It's a detector that completely redefines how we capture light, giving us this new kind of eye. Unlike your standard digital camera, or even the fancy scientific ones like SCMOS or EMCCD detectors, which grab a whole frame of light intensity at fixed intervals, event cameras work on a totally different principle. They don't really care about the absolute brightness level across the whole sensor.
Okay, so what do they care about then, if not the overall brightness?
They care about change.
Sure.
So traditional frame-based cameras record I, the intensity snapshot. Event cameras respond to I, the change in intensity, at each individual pixel.
Ah, okay. So instead of one big shutter for the whole scene, it's like millions of tiny independent light switches.
That's a great analogy. Each pixel acts like its own switch. It only flips and records an event when the light hitting that specific pixel changes significantly, either gets brighter or dimmer. It's not constantly measuring and reporting the light level everywhere, just reporting the changes when and where they happen, asynchronously.
Asynchronously, meaning each pixel does its own thing, not waiting for the others.
Precisely. And this change detection approach, how does it actually lead to better imaging for these faint, fast, single-molecule blinks? Well, each pixel continuously monitors the brightness, but specifically, it tracks the logarithmic intensity.
Logarithmic, why is that important?
It's crucial for dynamic range. Biological samples can have incredibly bright spots right next to very faint signals, like our blinking molecules. A standard camera might get saturated by the bright parts or miss the faint parts. By responding to logarithmic changes, the event camera is sensitive to small changes in dim light and big changes in bright light. It handles the range much better.
Got it. So it sees the whispers and the shouts equally well in terms of change.
Exactly. And when that logarithmic change crosses a set threshold, light got brighter or dimmer enough, the pixel instantly fires off an event. It doesn't wait for a frame signal. This event package is super precise. It contains the pixel's XY location, the exact time t down to microseconds, and the polarity p, was it a +1 brighter or -1 dimmer change?
Microsecond timing. Wow. So what are the big advantages that come from this way of working?
Several huge ones. First, asynchronous detection. Each pixel is independent, no waiting. Second, incredibly high temporal resolution. We're talking effective frame rates equivalent to maybe 100,000 frames per second if you think about the continuous data stream, way beyond traditional cameras.
Okay, that's fast.
Third, and this is critical, negligible readout time. In a normal camera, after exposure, you have to wait while the whole chip reads out the data. Time is lost, photons are missed. Here, there's no frame readout delay. The camera is always ready to detect the next change. No lost photons, no dead time waiting for the camera.
So it captures everything continuously.
Pretty much. And finally, near-zero motion blur. Because it reacts instantly to change, a fast-moving molecule doesn't get smeared out over a long exposure. The event marks its position at that precise moment of change.
That makes sense.
The source material has a great illustration, like their Figure 1 conceptual diagram. Imagine flashing a laser on and off. A normal camera sees a smooth hump of light during the on time. The event camera, it just sees sharp spikes. Red spikes when the light turns on, intensity increase. Blue spikes when it turns off, intensity decrease. Only the change gets recorded.
Ah, okay. Visually distinct events marking the start and end.
Right. It shows we're not bound by artificial frame rates anymore. We're capturing the actual events of light emission as they happen. It's like going from that flip book with missing pages to a seamless high-def movie.
A seamless movie. I like that. Okay, so we have this amazing camera that sees change with incredible speed and precision. How does Event SMLM use this to actually pinpoint a molecule's location?
All right, that's the next piece. We know single molecules do that characteristic blinking. They emit photons for a bit, then go dark, randomly.
Very event-driven process itself.
Exactly. Traditional SMLM catches some photons during the on time, but integrates them over its frame duration, maybe 30 milliseconds or so. That can blur the exact location and definitely misses any dynamics within that blink.
But Event's SMLM?
Event SMLM superpower is that within that same, say, 30 millisecond on time, it can capture multiple snapshots of the molecule's light emission because its own shutter speed is so much faster. It collects events over much shorter intervals, like just 3 milliseconds. So from one blink, it gets several rapid mini-bursts of photon events. These represent multiple realizations of what they call the temporal point spread function, or TPSF, basically, the cloud of photons detected from the molecule during that short 3 mils window.
So instead of one blurry picture per blink, you get several sharper data points from that single blink.
Precisely. It gives us a much richer data set for each molecular flash, more information to work with.
Okay, so you have this stream of incredibly timed photon events from these mini bursts. How do you turn that into one super precise location? You mentioned geometric localization.
Yes, and this is really clever. Events SMLM uses this geometric localization method, which is quite different from the usual way SMLM calculates position. Often, standard SMLM tries to fit a mathematical model, like a Gaussian shape, to the blurry spot of light. But that assumes the light spot is a perfect Gaussian, which isn't always true, and noise can mess it up.
Right, models have assumptions.
Event SMLM takes a more direct, data-driven approach, uses the actual pattern of detected photon events.
How does that work step by step?
Okay, so first, you collect the events over those short intervals, say 3 milliseconds, getting multiple TPSFs from one blink. Think of these as little clouds of photon detections. Second, for each of these little clouds, you calculate its centroid, basically, the center of mass of the light detected in that 3 meters window. This gives you several individual position estimates from the molecule during its brief on time.
Multiple guesses for where it was.
Kind of, yeah. Multiple precise estimates. Third, because these centroids all came from the same blinking molecule, they should cluster together spatially. So you mathematically fit a circle through these centroid points.
Fit a circle to the cluster of estimates.
Yeah.
Okay.
Fourth, the center of that fitted circle gives you your final, highly accurate position estimate for the molecule. It's averaged robustly from those multiple instantaneous readings.
And how do you know how precise that final position is?
Good question. Fifth, they associate a Gaussian profile with that fitted circle, and the width of that Gaussia, the full width at half maximum, or FWHM, directly tells you the geometric localization precision. And here's the key. This precision isn't based on a theoretical formula like in standard SMLM. It's purely experimental, derived directly from the spread of those calculated centroid points. It's a direct measurement of how well-defined the molecule's position is based on the actual detected photons.
So it's grounded entirely in the data you actually collected.
Exactly. No theoretical assumptions about photon counts or background noise needed for the precision estimate itself. Plus, the event camera's nature-only recording changes above a threshold inherently helps filter out steady background noise, giving cleaner data to start with.
That sounds much more robust. And you mentioned a new metric, PAR.
Yes, particle resolution, or PAR. It's a way to capture both space and time improvements in one number. It's defined as 2.3 times the localization precision times the collection time. The units are par, where 1 par is 1011 meter-seconds. Lower PAR means better spatiotemporal resolution. You can distinguish particles that are closer together and moving faster.
So it quantifies the molecular movie quality, in a sense.
In a way, yes. It pushes researchers to think about optimizing both aspects together. This whole geometric localization, combined with the event camera speed, it's like doing super-fast triangulation on a moving, nanoscale target. It gives unparalleled detail on position and dynamics.
That geometric approach sounds really powerful because it's so data-driven. But, I mean, are there situations where it might struggle, like if a molecule blinks really dimly or super fast?
Yeah, it's a fair point. Like any method, it has its optimal conditions. If a molecule emits very few photons during its on state, or if that on state is incredibly short, you might not get enough distinct events to calculate multiple reliable centroids and fit that circle accurately.
So you need a certain number of photon events per blink to make it work well.
You do, yes. But the sensitivity of the event camera often helps capture even faint photons. And crucially, the researchers didn't just rely on theory. They really put Event's SMLM through its paces on real samples to show it works.
Let's get into that evidence then. Does it actually deliver these amazing improvements in practice? What did they test it on first?
They started with a classic benchmark, scattered C3I dye molecules fixed on a glass cover slip. Simple, but a standard test. Using Event SMLM with a tiny 3 millisecond collection time, they achieved an average localization precision of just 4.581 nanometers. It's hard to even picture how small that is.
4.5 nanometers. And how does that compare to the standard approach?
On the exact same sample, standard SMLM, using a good scientific camera, an EMC-CD, only got about 34.6 nanometers precision.
Wow, so 4.5 versus 34.6, that's roughly a seven-fold improvement in precision.
Exactly, 7 times sharper localization. And the PAR value, that spatiotemporal metric, was 3.16 par for Event SMLM, significantly better than what standard SMLM could achieve. If you look at their figure 2, the visual difference is just night and day. The Event SMLM image, Fig 2b, shows these crisp, distinct dots, while the standard SMLM image, Fig 2c, is much blurrier. You can see the improvement.
Okay, impressive on a simple dye, but what about something more complex, like inside a cell?
Right, the next step. They imaged the mitochondrial network in fixed cells. Specifically, they looked at a protein called TOM20, which is embedded in the outer mitochondrial membrane, using a label called MEOS.
Mitochondria, the powerhouses, so seeing their structure clearly is important.
Very important for understanding cell energy, metabolism, certain diseases. Here, with Event SMLM and a 10 millisecond collection time, they hit 5.677 nanometers localization precision.
And standard SMLM.
Standard SMLM on the same mitochondrial network gave about 40 nanometers precision. So again, that's roughly another seven-fold improvement for Event SMLM.
Consistently better. And visually.
Visually, it was striking. The clusters of TOM20, which can look a bit like diffuse blobs with standard methods, became much sharper, more defined. You can see finer details in the network structure. Their figure 3 points out specific regions, R1, R2, R3, where Event SMLM, Fig 3b, clearly resolve structures that are just blurred together in the standard SMLM image. Fig 3c. You really see the intricate architecture pop out.
Okay, fixed cells look much better, but the real promise was dynamics, right? Capturing the movie in live cells. Did they do that?
Ah, yes. This connects right back to the core goal, moving from static pictures to life in motion. This is where Event SMLM, I think, truly blows the doors open. It forces you to ask, what can we learn when we track dynamics at this level? Their third test case is the most exciting, imaging hemagglutinin HA clusters in live NIH3T3 cells. HA is a key protein from the influenza A virus.
Influenza. So this is directly relevant to understanding viral infection.
Absolutely. Understanding how HA proteins move, cluster together, and interact with the host cell on the membrane is critical for figuring out how the virus gets in and replicates. In these live cells, Event SMLM achieved around 6.138 nanometers precision, again with a fast 10 millisecond collection time.
10 milliseconds. How does that compare speed-wise to standard SMLM in live cells?
That's a huge gain. Standard SMLM might need 60 milliseconds or more per frame just for exposure, plus readout time. So Event SMLM here is roughly 6 times faster in acquiring each data point or effective frame.
6 times faster acquisition. That's crucial for capturing fast events.
Critically important. And the PAR value, an impressive 14.11 par. That's about 17.8 times better spatiotemporally than standard SMLM in this demanding live cell scenario.
Nearly 18 times better spatiotemporal resolution.
But numbers aside, the real breakthrough was the dynamic observation. For the very first time, using this event-based approach, they could visualize the temporal dynamics, the actual movement, clustering, and unclustering of these HA proteins in live cells continuously for a full 60 minutes.
60 minutes continuously. Wow. So not just snapshots, but the whole process unfolding.
Exactly. A live molecular movie of viral protein behavior over a biologically relevant timescale.
How did they even analyze that much dynamic data? It must be incredibly complex.
They used a smart clustering algorithm called DBSCAN. It's good for SMLM data because it finds clusters based on density without you having to tell it how many clusters to look for or what shape they should be.
Okay, makes sense. What did the analysis show?
First, they confirmed it was reliable. The clusters found by event SMLM matched well, over 81% colocalization, with those found by standard SMLM, just much sharper and faster. But the dynamic analysis was the key. Looking at changes every 15 minutes, they saw the total area covered by HA clusters growing, the number of HA molecules in each cluster increasing, and generally more HA participating in clusters over time after the cells were given the HA gene.
So they could quantify the assembly process.
Yes, and visualize it directly. They could literally see clusters growing marked with arrows in their figures, sometimes multiple arrows for faster growth. They saw clusters shrinking or disappearing, and new ones forming out of nowhere. Figures 6 and 7 are amazing. They show these time-lapse images where cluster growth is color-coded, like orange, red, brown, and decline is blue. They tracked every cluster. Did it stay, disappear, or just arrive?
That's incredible detail. You're literally watching the virus components assemble and rearrange themselves on the cell surface in real time.
Precisely. It's not just clearer pictures, it's seeing the biological events unfold. For virology, understanding these assembly and trafficking steps is fundamental. Watching the molecular movie like this instead of static photos is transformative for figuring out infection mechanisms and potentially finding new ways to stop them.
That live cell data really drives home the power. Okay, stepping back, what does this all mean for the big picture? What are the core takeaways?
Well, the advantages are clear now, I think. You get superior spatiotemporal resolution, seeing smaller and faster. You have that negligible readout time, so you capture more of the signal without gaps. And you have this purely data-driven geometric localization, which gives you a more robust, experimentally grounded measure of precision.
So we can finally resolve individual molecules and track their rapid movements and interactions in real time, watching life's machinery actually work.
That's the essence of it. It's a monumental leap.
Now, you mentioned earlier, no technology is perfect. What are the current challenges or complexities with these event cameras that researchers are still working on?
Right, it's important to be balanced. The source material mentions things like latencies, tiny delays in reporting events. There are refractory periods. After a pixel fires, it needs a tiny moment to reset, like a neuron, creating a brief dead time. And there's always quantization noise, small errors, when converting the analog light signal to digital values.
So ongoing refinements are needed?
Definitely. But the field is moving incredibly fast. People are engineering brighter, more stable fluorescent probes, the blinking lights we track. And the event cameras themselves are getting better, particularly in quantum efficiency, meaning they capture a higher percentage of the photons that hit them. These ongoing improvements are constantly pushing the limits, making events SMLM more powerful and easier to use.
It sounds like the potential is just beginning to be tapped.
Absolutely. And what's really fascinating is how the researchers behind this work see the future. They don't just view event SMLM as an upgrade. They envision the emergence of a whole new kind of microscopy. They think these event-based systems could push that spatiotemporal PAR value into the sub-10 par range, maybe even below 1 par eventually.
Sub-1 par, what would that even allow us to see?
Imagine tracking almost every twist and turn of a single protein molecule in real time inside a living cell with near-perfect accuracy. It's mind-boggling. Which really raises an important question for you listening right now. What biological mysteries, previously completely inaccessible, could you start to unravel if you had that kind of power? That ability to watch the molecular dance in such exquisite detail and speed.
Yeah, the possibilities seem endless.
Think about understanding disease progression at the most fundamental single molecule level. Watching a drug molecule actually find and bind its target inside a live cell, seeing the immediate effect. Or finally untangling the incredibly fast signaling cascades in our brains, literally following signals molecule by molecule. This tech promises to unlock secrets we couldn't even dream of asking about before. It feels like moving from reading static blueprints to watching dynamic, living narratives of how life actually works.
And that's our deep dive into Event SMLM, a truly exciting, honestly transformative development in microscopy. From breaking the old diffraction limit to now capturing the real-time dance of molecules in living cells. It's just a powerful reminder that the boundaries of what we can see and therefore understand are always expanding. It really challenges our ideas of what's possible. Thank you for joining us on this journey today into the nanoscopic world.
It really does move us firmly into an era beyond static snapshots, letting us see biology truly in action. So my provocative thought for you to mull over is this. As we gain this incredible ability to visualize the intricate real-time movements and interactions of single molecules inside a cell, the actual ballet of life, the mechanics of health, the subtle start of disease, how might this granular dynamic insight fundamentally shift our very understanding of health and disease? Could we move beyond broad observations to truly personalized molecular insights? Diagnostics and therapies tailored not just to a person, but to the specific molecular behaviors happening inside their cells. The potential for discovery feels immense right now. Keep exploring.